The world around us teems with life we cannot see. These are the microorganisms, microscopic organisms inhabiting every corner of the planet. While some microorganisms are beneficial, others, known as pathogens, can cause disease. Understanding these invisible threats is crucial for medical science and human health. Pathogens have shaped the course of history, causing devastating epidemics and shaping our immune systems. Today, scientists study pathogens to develop vaccines, antibiotics, and antiviral drugs. This constant battle against infectious diseases is a testament to the importance of understanding these microscopic foes. The fight against pathogens is a continuous one. New diseases emerge and old ones re-emerge with drug resistance. By understanding the mechanisms of infection, spread and treatment, we can better protect ourselves from these invisible threats. The study of microorganisms is not just about combating disease, it's about understanding a fundamental aspect of life on Earth. Pathogens, the agents of infectious diseases, are a diverse group of microorganisms. These include bacteria, viruses, fungi, protozoa, and prions. Each type of pathogen has unique characteristics and mechanisms for causing disease. Bacteria, single-celled organisms, are responsible for illnesses like tuberculosis, pneumonia, and urinary tract infections. Viruses, even smaller than bacteria, are essentially packets of genetic material that hijack host cells to replicate, causing diseases like influenza, HIV/AIDS, and COVID-19. Fungi, a diverse group including yeasts and molds, can cause infections like athlete's foot, ringworm, and more serious systemic infections. Protozoa, single-celled organisms often found in contaminated water, can cause diseases like malaria and amoebic dysentery. Prions, misfolded proteins, are responsible for rare but fatal neurodegenerative diseases like Creutzfeldt-Jakob disease. Understanding the specific characteristics of each type of pathogen is crucial for diagnosis, treatment, and prevention of infectious diseases. By studying their life cycles, modes of transmission, and mechanisms of causing disease, we can develop targeted strategies to combat these microscopic threats. Bacteria are single-celled organisms that can survive in diverse environments, both inside and outside the human body. They are prokaryotes, meaning they lack a true nucleus, and they reproduce asexually through binary fission, dividing into two identical daughter cells. Bacteria are classified based on their shape, staining properties, and genetic makeup. Some bacteria are harmless or even beneficial, like those found in our gut that aid in digestion. Others, however, are pathogenic, possessing virulence factors that allow them to invade tissues, evade the immune system, and cause disease. Bacterial infections are typically treated with antibiotics, drugs that target specific bacterial structures or processes. However, the overuse and misuse of antibiotics have led to the emergence of antibiotic-resistant bacteria, posing a serious threat to global health. Understanding the mechanisms of antibiotic resistance is crucial for developing new drugs and strategies to combat these superbugs. Additionally, promoting responsible antibiotic use and infection control measures are essential for preventing the spread of resistant bacteria. Viruses are much smaller than bacteria and are not considered true living organisms. They're essentially packets of genetic material, either DNA or RNA, enclosed in a protein coat. Viruses lack the cellular machinery to replicate on their own and must invade host cells to reproduce. Once inside a host cell, the virus releases its genetic material, which hijacks the cell's machinery to produce more viruses. These new viruses then burst out of the infected cell, often destroying it in the process, and go on to infect other cells. Viruses are responsible for a wide range of diseases, from the common cold to more serious illnesses like HIV/AIDS, Ebola, and COVID-19. Transmission routes vary depending on the virus and can include respiratory droplets, bodily fluids, contaminated food or water, or insect bites. Antiviral drugs can help to inhibit viral replication but are often specific to certain viruses. Vaccines, which stimulate the immune system to recognize and fight off specific viruses, are one of the most effective ways to prevent viral infections. Section 5. Fungi, the spore-bearing threat. Fungi are a diverse group of eukaryotic organisms that include yeasts, molds, and mushrooms. They are found in various environments and play important roles in decomposition and nutrient cycling. However, some fungi can cause infections in humans, ranging from mild skin infections to serious systemic diseases. Yeasts are single-celled fungi that reproduce by budding, while molds are multicellular fungi that grow in long filaments called hyphae. Fungi reproduce both sexually and asexually, producing spores that can disperse widely in the environment. Fungal infections, also known as mycoses, can be classified as superficial, subcutaneous, or systemic. Superficial mycoses affect the skin, hair, and nails, while subcutaneous mycoses involve deeper layers of skin and underlying tissues. Systemic mycoses affect internal organs and can be life-threatening, especially in individuals with weakened immune systems. Diagnosis of fungal infections often involves microscopic examination of clinical specimens, such as skin scrapings or tissue biopsies. Treatment typically involves antifungal medications, which target specific fungal structures or metabolic pathways. Section 6. Unmasking the enemy, identifying pathogens. Identifying the specific pathogen causing an infection is crucial for effective treatment and control measures. Clinical laboratories employ a variety of techniques to detect, isolate, and identify pathogens from patient samples. One common method is microscopy, where specimens are examined under a microscope to visualize the shape, size, and staining characteristics of microorganisms. Culturing involves growing microorganisms on artificial media to observe their growth patterns and biochemical properties, aiding in identification. Biochemical tests assess the metabolic capabilities of microorganisms, providing clues to their identity. Serological tests detect the presence of antibodies or antigens specific to certain pathogens in patient serum. Molecular techniques such as polymerase chain reaction or PCR amplify specific DNA or RNA sequences, allowing for rapid and sensitive detection of pathogens. Accurate pathogen identification guides appropriate treatment decisions, facilitates epidemiological surveillance, and informs public health interventions to prevent further spread of disease. Section 7. A World of Factors: How Diseases Spread. The spread of infectious diseases is a complex interplay of factors involving the pathogen, the host, and the environment. Understanding these factors is crucial for developing effective strategies to control and prevent outbreaks. Pathogen factors include virulence, which refers to the ability of a pathogen to cause disease, its mode of transmission, and its ability to survive outside the host. Host factors encompass age, immune status, nutritional status, and underlying health conditions. Environmental factors play a significant role in disease transmission and include factors like sanitation, access to clean water, climate change, and population density. Socioeconomic factors, such as poverty, overcrowding, and lack of access to healthcare, can exacerbate the spread of infectious diseases. Globalization and increased travel and trade have interconnected the world, making it easier for diseases to spread rapidly across borders. Climate change can alter the geographic distribution of vectors, such as mosquitoes, leading to the emergence of diseases in new areas. Section 8. Fighting back, prevention and control. Preventing and controlling the spread of infectious diseases requires a multifaceted approach, involving individual efforts, public health interventions, and global cooperation. Vaccination is one of the most effective ways to prevent infectious diseases. Vaccines stimulate the immune system to recognize and fight off specific pathogens, providing long-lasting protection. Public health measures such as sanitation and hygiene programs, access to clean water, and vector control are essential for preventing disease transmission. Surveillance systems are crucial for tracking the emergence and spread of infectious diseases, allowing for early detection and rapid response. Antimicrobial stewardship, which promotes the responsible use of antibiotics, is essential for combating antibiotic resistance. Health education campaigns can empower individuals to make informed decisions about their health, such as practicing good hygiene, seeking appropriate medical care, and getting vaccinated. International collaboration is vital for sharing information, coordinating responses, and developing new tools and technologies to combat infectious diseases. Section 9. Tropical troubles, unique challenges. Tropical countries face unique challenges in controlling infectious diseases due to a combination of factors, including climate, poverty, and limited healthcare infrastructure. The warm, humid climate of the tropics provides ideal conditions for many pathogens and vectors to thrive. Vector-borne diseases such as malaria, dengue fever, and Zika virus are major public health concerns in these regions. Poverty can exacerbate the spread of infectious diseases due to factors like overcrowding, poor sanitation, and limited access to clean water and healthcare. Malnutrition and underlying health conditions can weaken immune systems, making individuals more susceptible to infections. Building robust healthcare systems, strengthening disease surveillance, and implementing effective control measures are crucial for addressing the burden of infectious diseases in tropical countries. International partnerships and support are essential for providing resources, expertise, and capacity building. Section 10. Safety first. Handling pathogens. Working with pathogens in a laboratory setting requires strict safety protocols to prevent accidental exposure and potential outbreaks. Laboratory personnel must adhere to standard precautions, which include treating all biological materials as potentially infectious. Personal protective equipment, or PE, such as gloves, lab coats, eye protection, and respirators, must be worn appropriately to minimize the risk of contact with infectious agents. Proper hand hygiene using soap and water or alcohol-based hand sanitizers is crucial before and after handling specimens. Safe handling and disposal of sharps, such as needles and scalpels, are essential for preventing needle stick injuries. Decontamination procedures, including disinfection and sterilization, must be followed rigorously to eliminate pathogens from surfaces and equipment. Laboratories handling highly infectious or dangerous pathogens require higher levels of biosafety containment, with specialized facilities and engineering controls to prevent the release of pathogens into the environment. Ongoing training, education, and adherence to safety protocols are essential for ensuring a safe working environment and protecting public health.